Hello students. So in this video we are going to study about the post-transcriptional processing. In the previous videos we have studied how the transcription is done with the help of RNA polymerase. Okay, and we have studied कि eukaryotes have split gene arrangement. Split gene means introns are present along with exons. Okay, so they are having introns and they are having exons also. Both are present, we call it as a split gene. And we know that RNA polymerase II will prepare hnRNA. If you remember, we have already discussed that eukaryotes have three types of RNA polymerase, 1, 2, and 3. And RNA polymerase II will prepare hnRNA. hnRNA means heterogeneous nuclear RNA. It is also known as precursor of messenger RNA. Okay, it is also known as precursor of messenger RNA. So I think so this is clear कि first of all basic you should know कि why we are studying this topic. We are studying because eukaryote genes are split genes. Okay, they have a split gene arrangement and we are using three type of RNA polymerase and our RNA polymerase 2 will prepare hnRNA. Why it is preparing hnRNA? Reason is again the same split gene arrangement. So because of the split gene arrangement, they are preparing hnRNA. They can't directly prepare messenger RNA. Now see here, this is your DNA and RNA polymerase 2 is preparing the primary transcript. This is the primary transcript which is prepared, which is of the same length as of the DNA, and this primary transcript is known as hnRNA. Okay, first of all, this is clear. Now, inside this hnRNA, just see this is area. Suppose this area is introns. Okay, I'm using the green color for introns. So these are your non-coding areas. You know very well, introns are your non-coding areas. So these are your introns. So this is your intron 1, this is your intron 2, and this is your intron 3. As a result, this area which is left here, this is your exon. We will say exon 1, exon 2, exon 3. Okay, and this is your exon 4. So we have to remove now, first of all, our step is we have to remove these introns. Now, before removing these introns, what we will do, see the board carefully, we want to make it as a fifth end. So here we will add, see here this side, we will add guanosine triphosphate. I've already discussed what is guanosine in the previous videos. Guanosine is your nucleoside. Okay, guanosine is nucleoside. With one phosphate we say nucleotide, but this is three phosphates are used, so we will say guanosine triphosphate. We will not use the word nucleotide here, which is methylated at 7th position. At 7th position, methyl group is attached to it. And we say we have done the capping work. So capping is done. This is your unusual nucleotide which is attached at one end, and the end which is attached, that end is known as now fifth end. Is it clear? This is now known as which end? We call it as fifth end. And the process is known as capping. Now see, after the capping work is over, in the meantime, check the board carefully, in the meantime, your small nuclear RNA protein will attach at the starting of the introns and at the end of the introns. See here, they will attach at the starting and they will attach at the end of the introns. What are these? These are known as small nuclear RNA, which is bind with the protein. So we say small nuclear RNA protein. Okay, which is also known as SNRP, which is helping in the process in which we have to remove the introns, and this process is known as splicing. Now, as a result, what will happen? See the board carefully. your snub will come near. Okay, see the board, how I will draw the diagram. Understand it carefully. And these your introns will come out in the form of a loop, because two snub will be brought near. As a result, the introns are coming out in the form of a loop. So these loops are known as spliceosomes. And this process in which we are going to remove the introns is known as splicing. Is it clear? The process in which we are going to remove the introns is known as splicing. In the meantime, what will happen? See the board. This is your area opposite to the fifth end, it is third end. In a template independent manner, we are not using any template and we are adding poly A. So poly A is added and we call this step as tailing. This is tailing is done. Okay, so tailing is done, and tailing is done to make it as a third end. So this is identified as third end, and this, so we have already studied that this is identified as fifth end. So your first step will be capping. Understand the board carefully. First step will be capping. Second will be tailing. Till now, if you check carefully, I have not removed the spliceosomes. Now, in this final step, we are finding splicing is done. Okay, so splicing is done, completed. Though it started while they were doing the tailing, the time only started, they made the spliceosome, but completion is done after the tailing is done. Okay, so your third step will be your splicing. Now this RNA which is formed, now this is known as mature messenger RNA. So now this messenger RNA is now known as mature messenger RNA. I hope this topic is clear to you. Now see the entire summary of this. If you know, this was DNA, and RNA polymerase made hnRNA, which has gone first for capping, then tailing, and during that time, the introns are removed. This green color were introns. Okay, so introns are removed with the help of a small nuclear RNA protein, and third step will be splicing, and this structure which is formed now, it is known as mature messenger RNA. And if you check the diagram carefully, you will find this mature messenger RNA is shorter than the hnRNA. Okay, so this mature messenger RNA is shorter than this one. See the board carefully, this one. Okay, this diagram which is drawn here. So this is your shorter than this one. Okay, it is shorter than your hnRNA. Why? Because here introns are removed and it's having only and only exons. Now it is ready to go out and do the process of the translation, which we'll be discussing in the next video. I hope this video will help you to improve the topic. Thanks for watching.
If you liked it, please give it a thumbs up and subscribe to my YouTube channel.